In this lesson, we'll be looking at digestive enzymes. So we have a few images on this screen over here, and we'll go into them in a bit more detail in a moment. But the first thing that we need to be able to do is actually define what an enzyme is. So on this screen here, we've got a few coded sentences. So what you need to do right now is pause the screen and see if you can decode each one. Once you've done these two, have a go at doing the secure task as well. Right, so let's go through the answers. What is an enzyme? An enzyme is actually a biological catalyst that speeds up the rate of reaction. So you probably might have heard of catalysts in chemistry. So catalysts generally speed up reactions. Enzymes are the same, but they're just biological because they're found in our bodies, so they're living, yeah? Enzymes are also made of proteins. So that's what the actual structure is made up of. It's made up of proteins, not fats, etc. It's proteins. And then the secure is that digestive enzymes are actually made in the pancreas. So the pancreas, I suppose, is probably the most prevalent in the digestive system for production of enzymes. We do have an enzyme called amylase produced in the saliva, and in the stomach as well. We've got pepsin, but the main ones are in the pancreas. So let's go back to these images and let's try and explain what they mean. So the top ones are to do with the lock and key theory. So the lock and key theory states that with an enzyme, you always have a specific substrate that can bind. So for example, in this image over here, only this shape of substrate will be able to fit into this active site for the enzyme. No other substrate will be able to get in. So if we had like a square shape, that would not be able to fit into this enzyme. So once this substrate binds to the enzyme, we form an enzyme substrate complex. So for example, if this was a protease, which is an enzyme that breaks down proteins, and this is a protein, they'll kind of match and the shape will be complementary to the enzyme. So they'll form this complex. Once the complex has been formed, the enzyme can do its job, and the enzyme can literally break down the substrate into the product. So what we're trying to do is we're trying to break down the protein into something that's more soluble and easier for our body to work with. So with proteins, we want to break them down into amino acids. Once that process has been done and the enzyme has catalyzed our substrate, we end up with products. To sum up what I just talked about, so the fact that enzymes are specific, substrates will bind to the active site, we form the complex. I don't think I mentioned this, but bonds are actually made or broken depending on kind of what we're trying to do with the substrate. And then obviously products are released. Next, we need to know what's going to happen to an enzyme if it becomes denatured. So when we're talking about denatured, we literally just mean destroyed. The enzyme is no longer functioning. So for example, if we look at this side of the diagram here, we've got some heat. Now heat can be good only up to a certain point, because if an enzyme can't function in kind of really high temperatures, what will happen is that it does actually become denatured. So what happens is that the temperature is too high, so the active site actually changes shape, and because the active site changes shape, as you can see here, the substrate can no longer bind, and then it can't form the enzyme substrate complex, and therefore you can't end up with your products either. So this is a graph which shows that quite well. I'd probably say 40 degrees is the optimum temperature for this enzyme, whichever one it is. That's because if you look at the peak of the kind of graph, it functions the best at about 40 degrees, so we call that its optimum. Below that, it doesn't function very well, but as we keep increasing the temperature, the enzyme activity does increase, but only up to this point. Once we carry on increasing the temperature too much, that's no longer the optimum, and that's too high, and that's denatured our enzyme, so our active site has changed shape now. Here's a little exam question that we're going to go through. So we need to figure out what the optimum pH is for the catalase enzyme. And we also need to explain what has happened at pH 1 and at pH 14. If you want to read the question and have a go at giving an answer, you can do so now. Just pause the video. Okay, let's go through it. So it says that the student carried out the investigation to study the effect of pH on catalase. In the presence of catalase, hydrogen peroxide breaks down to release oxygen gas. So we know that's what we're looking for. We're trying to see hydrogen peroxide breaking down, and we're trying to see lots of oxygen gas bubbles. So we've got 5 tubes here, and they observe the amount of oxygen gas released. So automatically I can see that the most bubbles are found in pH 7. That must mean that the optimum pH for catalase is actually pH 7, because that's when we saw the most hydrogen peroxide break down into oxygen gas. On the other hand, what has happened at pH 1 and 14, we've got absolutely no bubbles released over here. So that must mean, if you look at, pH 1 is very, very acidic if you look at the pH scale, and pH 14 is very, very alkaline. So there is a chance that these very low pHs or very high pHs may have denatured our enzyme, and that's why we see no breakdown of hydrogen peroxide into oxygen gas. Whereas in the kind of middle ground over here with pH 5 and pH 9, we did see some breakdown, but here, none at all. We need to also know about digestive enzymes. So again, if you want to just pause the video here, decode these 2 sentences, and also try and explain why we don't just have one enzyme to catalyze every substrate. Let's go through the answers. So the first one should have been this: convert food into small soluble molecules that can be absorbed into the blood. So that's what digestive enzymes do. It's all about getting sort of nutrients from our food and turning it into a form that our body can kind of accept. Then the molecules are used to build new carbohydrates, lipids, and proteins. And finally, the secure task: every enzyme is specific to its substrate. So that's why we can't just have one enzyme that matches every single substrate. The substrates are going to be different, so therefore our enzymes have to be different as well. They have to have different active sites for each substrate. Finally, let's go through the three different enzymes that we've got that we're going to work with for digestive enzymes. Firstly, we've got carbohydrases. It's in the name. They break down carbohydrates into simple sugars. The one that I mentioned earlier was amylase, and that's a carbohydrate that can actually break down starch. So it gets the starch molecule, breaks it down into little sugar molecules or glucose molecules. We've also got protein, which we mentioned at the start of the video, which protease enzyme, which breaks down proteins into amino acids, which are then soluble. And finally, we also have lipids. So lipids is the scientific term for fats and oils, and we break them down using this lipase enzyme, and they turn into fatty acids and glycerol.